Okay, hello everyone, and welcome to today's presentation. What about the bugs? ABMI wetland monitoring, where we are at, how we got here, and where we are going with Rob Hinchlet. Rob is the lead scientist for aquatic invertebrates at the Alberta Biodiversity Monitoring Institute. Since 2006, he has been an integral part of the ABMI, starting as a field technician in the prototype year, where he helped test and refine the aquatics field protocols. Rob then coordinated fieldwork at the ABMI Monitoring Center for 2 years before joining the ABMI Processing Center at the Royal Alberta Museum in 2008. At the museum, he managed ABMI specimens across all taxonomic groups while honing his expertise in aquatic invertebrates. He is certified by the Society for Freshwater Science and Western EPT, general arthropods and chironomids, and most of his time these days is spent either gazing through a microscope and identifying any aquatic bugs that come his way, or dabbling in the data. Rob's most recent move was to the University of Alberta, where he currently resides, at least during the day, in the ABMI Aquatic Invertebrate Lab. His fascination with the diversity of aquatic bugs began early, shaping his professional journey and personal interests. Rob enjoys exploring all aspects of aquatic invertebrates and aquatic ecosystems, from fly tying and fly fishing to photographing aquatic bugs in natural settings. Beyond work, Rob also finds joy in backpacking, mountain biking travel, and exploring off the beaten path places, reflecting his deep connection to nature and adventure. With that, please take it away, Rob.
Awesome. Thanks, Brad. Yeah, hey everyone. Thanks for tuning into the webinar here.
Two things of.
I see we have a fairly large group of people in attendance, which is pretty encouraging. I think it's great to see that there's a lot of people that are interested in wetlands in general, wetland biodiversity, and certainly what the ABMI is up to these days. Before we begin, I'd just like to acknowledge that we work and live on the traditional lands of the Cree, Blackfoot, Métis, and other Indigenous peoples who have called this land home for countless generations. We recognize the profound injustices and wrongs that have been inflicted upon these communities throughout history, and respectfully acknowledge the ongoing impacts of colonialism, displacement, and cultural erasure that continue to affect Indigenous peoples today. We honor their histories, their cultures, and enduring connection to the territory, and we express our gratitude for their stewardship of these lands. We commit to fostering understanding and healing, and working towards reconciliation in our interactions and engagements with Indigenous communities moving forward. So I would say that this presentation is probably long overdue. As you're going to see, working with the wetland data is not without its challenges. And I think that's resulted in slower than expected progress on the analysis side of things. And in addition, I tend to get wrapped up in just getting the work done rather than working on the engagement side of things. So I'm hoping moving forward, we can share more of what we're doing, I guess, in a more regular and timely manner. On that note, I'd just like to give a quick plug to the ABMI Wetland Atlas. If you're not aware of this, it's a reporting tool that we started up last year to share regular updated information on wetland monitoring in Alberta, and also what the ABMI is up to as far as wetlands go. So you can find the atlas on the ABMI website under online reporting. And our plan is to update it twice a year moving forward. So it's really worth checking out on a regular basis if you want to kind of stay in tune with what we're doing. So what are we going to cover today? Well, we're going to start by looking at the use of aquatic invertebrates as indicators of stream health. And the whole idea here is that I want you to develop an understanding of why stream biomonitoring has been so successful. Then we're going to move on to the challenges faced in adapting stream monitoring approaches to wetlands. And that will highlight some of the difficulties in doing so, and also highlight some of the limitations in the data. Then I'd like to consider the utility of the current ABMI data set, along with approaches aimed at enhancing aquatic invertebrate data collections from wetlands, which will hopefully allow wetland biomonitoring to be applied at smaller spatial and temporal scales. So a bit of historical context for you here as far as stream biomonitoring goes. So really, compared to wetland biomonitoring, stream biomonitoring has a relatively long history and a fairly well-proven track record. The initial concept was actually pioneered back in the mid-1800s, and then it was later refined by Colquitt and Marsan in the early 1900s. So this work was all related to sewage and industrial wastewater contamination. And you have to remember, at this time, there was really no treatment for, sewage and wastewater being dumped into the rivers. So it was these early pioneers who not only realized that this was a problem, but they also came up with a simple way to measure the impacts. So their design was very simple in the beginning, as pretty much as organic or chemical pollution increased, aquatic invertebrate groups that are more tolerant of pollution would increase in numbers, while the groups that are intolerant of pollution would actually decrease. So a pretty simple system to work with. And since those early days of stream biomonitoring, of course, there's been quite a few refinements in, data collection and analysis methods and that sort of thing. But that basic approach is still used today. So the whole basic approach still applies. And yeah, just a little side note here, if you're curious about who gets the credit for the earliest form of biomonitoring, as I was, you can trace it back to the Greeks, the Romans, and the Chinese, where there's indications in some of their ancient and historical writings that they would use the presence of specific wild plant species as indicators of soil condition. And then they would actually use that information to predict their farming success and which crops would grow best. I was just a little curious about that because I thought maybe wetland biomonitoring was the earliest form, but nope, it's the Greeks, Romans, and Chinese. Yeah, so the work of Colquitt and Marsan did result in some slow changes to the way wastewater was handled over the years. But it was really in the later part of the 1900s when an interest in protecting our watersheds became more prevalent that the use of stream biomonitoring really started to catch on. And here generally the focus was mainly on protecting water quality at its source. So that being our primarily headwaters, rivers, and streams. So if you look at the typical habitat in these headwaters and streams, we know that it's, cold, clear, well oxygenated water. The streams might get murky at certain times throughout the year, but generally you have that clear water running through these headwater streams. If you consider the substrates, they're generally a mix of gravel and cobble. And there's also extensive open areas between the rocks and the gravel. And so all of this surface area creates a very diverse habitat where aquatic invertebrates inhabit not just the upper surface of the rocks, but they also occupy those interstitial spaces between the rocks. And then additional stream habitat complexity is the result of kind of that riffle, run, pool configuration in most streams, where the pools serve as kind of a depositional area where they collect and control the movement of sediment and organic material throughout the stream. Yeah, I should note here too. So when it comes to stream biomonitoring, when they're collecting the samples, it's typically the riffles and sometimes the runs that are sampled. And the reason for that is you want to sample that consistent habitat within the stream reach so that the results or the assessment of the aquatic invertebrate community remains consistent as well. Okay, so contrast this with streams impacted by human activity on the landscape. And you'll see that changes in the surrounding terrestrial habitat can actually have a profound effect on the invertebrate community within the stream itself. So sedimentation is one stressor that commonly affects headwater streams. And the sedimentation is generally the result of poorly controlled erosion, usually from things like construction, logging, agriculture, recreational activities, that sort of thing. So as sediment loads get washed into the stream, they have a number of detrimental effects on the whole stream ecosystem. The main one is they reduce light penetration into the stream itself, which leads to lower plant and periphyton productivity. It tends to smother the substrate, so it actually eliminates the surfaces on top where the periphyton grows. And if enough sediment builds up, it can also clog those interstitial spaces, and that reduces the livable habitat areas for a lot of the invertebrates. And then in general, the increased sediment loads, they pretty much stress out the organisms that are not really adapted to those higher levels of suspended sediments. So overall, the stream productivity and diversity is going to fall as the habitat becomes unlivable for those organisms that are adapted to the different microhabitats created by that original open matrix of cobble and gravel. Reduced forest cover on the landscape is another common stressor in a lot of watersheds. And so without the shading effect of a forest canopy, the soils across the surrounding landscape experience an increase in thermal loading. And so to picture this, if you think about the difference that you would feel standing in a mature forest versus an open meadow or bare ground on a hot sunny day, you can kind of get a feel for what the difference is when we have a clear-cut logging area. So as the rain flows through the surface layers on its way to the stream, that rainwater actually gets warmed up running through the landscape. And the difference in ground cover also results in changes to the nutrient profile of the rainwater entering the stream, because that rainwater percolates through those organic layers. And once you've removed the forest cover and you've altered those organic layers, it kind of changes the nutrient profile of water entering the stream. And so this all leads to a number of detrimental effects as well. So increased water temperature is one of the major ones. And with that increase in water temperature, you also have decreased oxygen levels. You're going to have changes to the flow and seasonal runoff patterns within the stream itself as well. And it can also result in changes to the algae species and growth rates within the stream system. So that paraphyton level that a lot of the aquatic invertebrates like to feed on can get replaced by more of those filamentous algae species that aren't really suitable for many of the organisms that do feed by scraping the paraphyton off the rocks. And then there's other external inputs that are also going to play a significant role in shaping the aquatic invertebrate community and streams. So you have things like changes to the riparian cover, which can affect the inputs of leaf litter and woody debris entering the stream, and both in terms of quantity and quality. So a good example of this is when streams the cover around a stream is replaced to help kind of control erosion if they've been doing any activities around the stream. So normally you might have species like aspen and river alder growing along the margins of the streams. And then in an effort to reestablish that buffer more quickly, they'll plant faster growing species, shrub species like willows typically. And that results in leaves shed into the stream that might not be usable by some of the native invertebrate species, or they may not provide the same nutritional value. And then, of course, there's urban runoff, organic pollution, and wastewater discharge that also play a role in stream health by introducing toxic chemicals, altering the water chemistry, and changing soluble nutrient concentration. So you'd have things like road salt, detergents, fertilizers, oils from poorly maintained vehicles that can all end up in the storm sewer system and consequently run off into a stream or a river. And then there are also chemical pollutants from industrial activities as well. So either you can have direct discharge or airborne chemicals that may be directly toxic to some of those sensitive aquatic invertebrate groups. So how is change measured? So most stream monitoring revolves around a metrics-based approach. So the actual counts of the taxonomic groups and the number of organisms in the sample are used to create different metrics that respond to changes in the stream ecosystem. So the basic measures like taxonomic richness and diversity are commonly used, but the most useful metrics are those that show a shift from sensitive taxa to more tolerant taxa. And generally, this is going to include EPT to some extent. So Ephemeroptera, Plecoptera, and Trichoptera. We just abbreviated as EPT. So that would be like your mayflies, stoneflies, and caddisflies. And these three groups are, pretty common in the headwaters, rivers, and streams, and they're also the most sensitive to change. And so any metrics that you see developed, a lot of them will involve EPT. Different functional feeding groups and tolerance values are also commonly used in the calculations. So as the habitat degrades, the invertebrate community will typically shift from that EPT dominated species community to being more made up of other more tolerant taxa like worms and dipterolarvae, and any other organisms that can tolerate those warm, low oxygen environments. So from here, the metrics are either used to calculate a single numerical value that represents the health of the stream. So we often refer to that as an index of biotic integrity, or an IBI for short. Or sometimes the metrics and or the actual aquatic invertebrate community data are subject to multivariate analysis, where you have these graphical outputs that show the relationships and patterns in multidimensional space. So you can see where you have the reference or the healthy sites typically cluster together, and then any impacted sites would tend to fall outside the zone. So the takeaway from all of this, as far as stream monitoring goes, is that in stream biomonitoring, those anthropogenic stressors on the landscape, they often result in significant and easily measurable changes to the aquatic invertebrate community. So we'll take a look at wetland biomonitoring here now. So compared to stream biomonitoring, the interest in monitoring and preserving wetlands really has a much shorter history. So even in the middle of the past century, it was thought that wetlands had no real recognizable value. And I would say that sentiment is still present to some extent even today, but I think we've come a long way. So hopefully the future holds something good for wetlands. So when it comes to wetland biomonitoring, it was initially thought that with that success of the stream biomonitoring, a similar rapid assessment approach would provide comparable results in wetland monitoring. But as we're finding here, it's proven not to be the case. If you look at some of the more recent work that's been done with wetland biomonitoring, there's been some success with a few localized studies, but those successes were usually looking at specific stressors within the environment. In general, though, many programs are really still struggling to relate wetland health to aquatic invertebrate communities in a broader context, especially when you're attempting to differentiate between individual wetlands that are impacted by human disturbance versus those that are not. So in order to understand why, we'll start by taking a look at the wetland habitat. So we know that wetland basins are more common at lower elevations, which are usually warmer, of course, or in areas of low topographical relief. And then, so being shallow by definition, those wetland habitats tend to warm up quickly in response to rising air temperatures. And that just results in warmer water temperatures compared to what you would see in headwater streams and rivers. The wetland substrates themselves, they're usually composed of that fine sediment or accumulations of organic material. And so that warmer water and the decomposition within the wetland will result in lower oxygen levels, and in some cases, hypoxia or even anoxic conditions within the wetland. If you look at wetlands in general, there's variable rates of productivity growth and decomposition between the wetland classes. So if you think of the difference between, say, a bog and a marsh, there's quite a bit of difference in, the decay rates and the productivity of those two types of habitats. And even within a single wetland class, you're going to see differences in productivity, water chemistry, and community structure, depending on where and how that wetland is actually embedded in the surrounding landscape. And then in addition to that, many wetlands have a complex heterogeneous habitat structure. So the wetland often exists as a wetland complex that may be made up of bogs, fens, marshes, all connected to that shallow open water. And then even within the shallow open water area, you can find a mix of bare substrates, emergent zones, submerged aquatic vegetation. And this could all exist as like a monoculture or mixes of different species. And it's all in kind of a discontinuous patchwork throughout the wetland. So wetlands are subject to similar stressors that we see in rivers and streams. I guess the exception would be generally agriculture replaces forest harvest operations in the prairie parkland region. But for the most part, they're subject to the same stressors on the landscape. So the same, human development and human activity that you would see up in the headwater streams. So I think, the impacts that are going to cause the greatest degree of change in streams they're muted in wetlands. So the increase in sedimentation that we saw in rivers and streams is going to have less of an effect on the wetland because the aquatic invertebrates that inhabit the wetland, they're already adapted to those depositional type habitats. Likewise, the increase in temperature is going to have less of an effect on the aquatic invertebrates in wetlands because a lot of the species that inhabit wetlands are already adapted to warm water and they're already adapted to lower oxygen conditions. Some of the factors that are expected to result in changes to aquatic invertebrate communities they're going to be limited to things like hydrology, nutrient inputs, climate change, chemical contamination, and that sort of thing. So yeah, so the EPT species that are most sensitive to change that we see in those upland rivers and streams, they're just not present in wetlands. Most of the taxa in wetlands, including some of the Ephemeroptera and Trichoptera species that are present in wetlands, they're certainly adapted to a broader range of conditions, and they can tolerate periods of warmer than normal temperatures, or even very low oxygen levels, either through physical or behavioral adaptations. And so they're well adapted to those types of environments. And even if you take like a lowland river or a stream, like what you would find around the Edmonton area here, you would also find that the responses to anthropogenic stressors are muted in those rivers and streams, because the aquatic invertebrate taxa that are present are already adapted to that silty, warm water conditions. So the takeaway from all of this is that when it comes to wetland biomonitoring, anthropogenic stressors are likely to result in subtle changes to the aquatic invertebrate community that, just due to the nature of the habitat and the organisms that live there, they're more difficult to measure. So in other words, the signal from wetland data is generally going to be much weaker than it is from rivers and streams. So yeah, so anytime you're trying to detect a weak signal, noise in the data becomes more of a problem. And that noise can come from multiple sources. And then, so collectively, it has the potential to mask any signal within the data. So some of the sources of variability or noise include the natural year-to-year variability in the community structure. And so that's driven by changing annual weather patterns, predator-prey interactions within the wetland, predator-prey interactions on the surrounding landscape. Yeah, so all of these things combined lead to differential breeding success. And so you have shifts in the aquatic invertebrate community from year to year. So that year-to-year variability is not really something that we have any control over, and it's something that's expected in most data sets. So what I want to focus on here are the other three sources of noise that we can exert at least some sort of control over. These would be seasonal variability, or what I like to call summer turnover, spatial variability within the wetland, which comes into play when you're collecting the samples, and then subsampling variability, where how the samples are processed in the lab can actually affect the final data. Yeah, so all of these sources of noise are going to affect our ability to create a data set that accurately represents the aquatic invertebrate community and the habitat being sampled, and also our ability to separate the signal from noise. So the seasonal variability speaks to the fact that wetland aquatic invertebrate communities are in a constant state of flux. So as we progress through the different seasons, the populations change in total and relative abundance. And there's a number of things going on that kind of cause the situation. So you're going to have aquatic invertebrates that are, eaten by other invertebrates and waterfowl. Organisms are completing their life cycle and dying of old age. We have eggs hatching into the next generation. We have mature larvae that are pupating or emerging into terrestrial adults. And then you also have early instar larvae that are becoming mature enough for us to actually identify them. So the rate of change from all of these interacting factors tends to be greatest in midsummer, as seen in the hypothetical graph at the top there. So the rate of change relates to the different taxonomic groups, either increasing or decreasing in numbers throughout the year. So you can see this play out in some of our general additive models for predicted abundance on the sample day. Yeah, I should note here, so when we're looking at the general additive models, the ABMI generally samples wetlands in midsummer, so that's between day 180 and 210, I guess, at the outside extreme. So if you look at the Anisoptera UID graph, this one represents early instar dragonfly larvae that we're just not able to identify beyond the suborder level because they're just tiny little first, second instar larvae. So you can see that they're present in low numbers in late spring and early summer, that kind of day 165 to day 180 range on the graph. And then as those eggs are hatching, the eggs that were laid earlier in the spring, they start to hatch. You're going to see a big influx of early instar larvae that build all the way into midsummer as more and more of those eggs hatch. And so you'll see the peak kind of in the middle of summer there is in terms of predicted abundance. And then as we move into late July and August, those larvae are starting to mature enough now that we can actually identify a greater portion of them to family. And there's also some predation at play here as well. So here we see the number of unidentified Anisoptera start to drop off. And then the other example we have there in the Enelagma graph to the right. So this represents damselfly larvae that we're able to identify to genus. And they start to emerge as adults early in June and continue emerging through to late June. So as this happens, you see the number of mature larvae that we can identify to genus decrease, and they reach their lowest levels in midsummer because they've all emerged and they're out, hunting, mating, and laying eggs. And then you'll start to see an increase in the numbers in late July and August when the larvae that have hatched out earlier on in June are starting to become mature enough that we can identify them to genus again. So these are the seasonal changes that we see in just 2 taxonomic groups. So if you can imagine this taking place across a few hundred groups, all during midsummer when the ABMI does its wetland sampling, and all at the same time, you can see that how there's all these different fluxes of changes in the population of the different aquatic invertebrate groups. So with all of that change happening over a short period of time, it becomes important that any repeat sampling we do at the same wetland would need to be done as close to the sampling date of the original sample collection as possible, because you want to be able to produce comparable results without that kind of confounding factor. And so that was certainly on our radar when we first created the sampling protocols when they were being developed. And then at that time, with the limited seasonal sampling data that we had available, we set a 14-day sample window for any wetlands undergoing repeat sampling. But we're starting to realize that the 14-day sampling window is likely too broad. And we're finding that even with the 14-day sampling window, it tends to be very difficult to keep the repeat sampling visits close to that original sampling date. So if you consider things like, scheduling multiple crews across the entire province, you've got a mix of first visit, second visit wetlands to be sampled in that summer. You're trying to coordinate days off, you're trying to coordinate site access, you've got last minute scheduling changes because of forest fire and weather delays and other unforeseen events. I think you can see how sticking to that tight repeat sampling window can be quite difficult to achieve. And even if the wetland is sampled on the same calendar day on subsequent visits, there would always be some variation due to differing degree days between the years. So the next source of noise I want to talk about is spatial variability. So patchy habitat and patchy distribution they're really nothing new when it comes to biological sampling, but I just want to show how this plays out when it comes to sampling wetlands. So we know that in any wetland, the habitat itself can be quite variable. So you might see, areas of emergent vegetation, submerged vegetation, you've got the bare substrates, and those are all interspersed throughout the wetland. And in addition, the submerged and the emergent vegetation can exist as areas of, different aquatic vegetation species, either coexisting as a mixed habitat, or they can form kind of a patchwork of monocultures. And so we know that the distribution of aquatic invertebrates within the wetland is driven by numerous factors, but habitat structure in the form of the physical surfaces and hiding places created by the vegetation is probably one of the biggest drivers of distribution of aquatic invertebrates within the wetland. But even in areas with homogeneous vegetation structure and homogeneous species composition, you're still going to see a significant variability in the number and ratios of organisms collected at different points. So you can see this in the short methods test that we conducted several years ago. So here we collected 10 samples using the ABMI triple sweep method along the shoreline of 2 different wetlands. Each net sweep was done in the same water depth range, so 45 to 60 centimeters deep, and the same habitat structure. So it was always 100% vegetation cover when we did this. So essentially you're sampling in places along the edge of the wetland that look identical to each other. So here we collected the 10 samples. And then the samples from each net sweep point were actually kept separate. And then we sorted them in their entirety. So that means that we didn't do any subsampling in the lab. We actually counted every organism in the sample. So you can see that even though the habitats were the same, the number of organisms collected varied widely between sample points, and the relative abundance of most groups also varied between the sample points. So of course, there's different options available to smooth out that variability. Collecting a sample along a continuous path or transect, something we call the traveling sweep net approach, or TSA, is one way to do that. You could combine multiple samples into a composite sample. We call this the composite transect approach, or CTA, and that's what the ABMI is currently using. But even with these two methods, excuse me, the data shows that neither of these approaches really managed to smooth out all of the noise. So in a paired sample test, where each sample was sorted to completion again, so we were controlling for subsampling noise in the lab, we saw clear differences in the total and relative abundance between the two samples. So the percent difference in total abundance varied between 1.6 and 60%. And the total cumulative percent difference in relative abundance at the core sorting level ranged from 30.1 to 110.6. So either method, the TSA or the CTA, generally shows a substantial amount of variability between the two samples collected at the same wetland on the same day. So we also have some additional unpaired data that suggests that the mean value for percent difference in the total abundance and total percent difference in relative abundance will converge as more sample data is added. So really that means that there's going to be no difference in variability between the CTA method and the TSA method. But we do need to get some actually get some more data on this before we can confirm that. So that's something that we're working on over the next few months here. So reducing spatial variability or noise could be further reduced by collecting more replicates, but then that's always adding to the cost and the time needed at the sites. So not always a good approach, especially when you're trying to sample a wetland in a single day. So as far as sorting samples in the lab goes, we know that sorting the complete sample is far too time consuming and expensive. And for that reason, samples are usually randomly subsampled in the lab. And that's done using a fixed count or in some cases, a percentage of the sample. And so you'd think that the numbers generated by random subsampling would be fairly consistent, especially if a reasonable proportion of the sample has been sorted. But we're finding there's a surprising amount of variability in the data generated from random subsampling. So a good way to understand this, why this is the case, would be if you can imagine the Marchant box containing exactly 100 marbles, and so that represents 100 bugs in the sample. And you flip that box and swirl the marbles in the lid of the upside down box, and then you flip the box back so that everything drops straight down into the cells. We call this a zero gravity flip in the lab. Because if you were to just simply rotate the box from the upside down to the upright position, all the heavy material, of course, and the bugs would end up in the near end of the box, and then the light material would flow to the far end. So the zero gravity flip uses gravity and momentum to kind of hold everything in the lid of the box until it's fully upright, and then all that randomized material just drops straight down into the cells. So we've got these 100 marbles in the Marchant box. We've done the zero gravity flip. And so with a random distribution like that, the probability of getting exactly one marble in each and every one of the 100 cells of the Marchant box is actually extremely low. So what is more likely is that some of the cells may contain one marble, some cells might contain no marbles, and some cells will contain two marbles. And then, of course, it's possible that some cells could contain, in order of decreasing probability, you could have 3, 4, 5, or more marbles just based on that random distribution. So if you were to randomly select a single cell, and that cell contained one marble, you would correctly conclude that the entire sample contains 100 marbles. But if you were to select a cell that contains no marbles, or maybe it contained 3 marbles, you would conclude that your sample contains either 0 or 300 marbles. If you take that a little bit further, you could say you randomly select 2 cells. One contains 1 marble, and then the second cell contains 2 marbles. you do the math and you conclude that your sample contains 150 marbles. So as you select more and more cells, the calculated value of the number of marbles in the Marchand box tends to get closer to the actual value, but it's always going to vary within a range of possibilities depending on which cells you randomly select. So yeah, let's see how this plays out with an actual bug sample. So this data is from a sample that was sorted to completion, meaning again, we sorted all 100 cells in the Marchand box to get the actual cell-by-cell counts and the total abundance for that sample. And then the actual cell-by-cell sorting counts were used to randomly resort the sample 10,000 times. So the normal distribution on the right there at the top shows the full range of possible outcomes for calculated total abundance in the sample. The vertical red line represents the actual total abundance that we counted in the sample. In this case, it was 4,236. And the vertical black lines represent plus or minus 5% of the actual abundance in the sample. So in the top graph, when sorting to a fixed count of 350 organisms, which is what the ABMI uses, the sorted total abundance will be within 5% of the actual total abundance less than half of the time. So if we were to expand the accepted error to plus or minus 10%, which represents a range of 850 in the count, there's still going to be a 1 in 4 chance that the calculated total abundance is going to be significantly different. And so as you sort more of the sample, say, and we sort to 750 fixed count in the bottom graph, those range of possible outcomes are going to tighten up. And so if you were looking at what counts were within plus or minus 5% of the actual count, now we're looking at it happening 55% of the time. So obviously, sorting a greater proportion of the sample provides a better estimate of the actual sample total. But of course, it also adds substantially more time and more cost to the sample processing. So total abundance is a metric that is known to be variable to the point that a lot of studies don't bother using it in IBI calculations. But we also wanted to look into how the relative abundance varied within the sample randomly. And so relative abundance is routinely used in both IBI calculations and community analysis. So using that same cell-by-cell sorting data, we again resorted the sample 10,000 times. We calculated the relative abundance or the percent of the sample of Ephemeroptera for each of the resorted samples. And you can see that the relative abundance shows that same variability pattern as the total abundance. And so that means that any community level analysis or calculation of IBI metrics is also going to have a certain amount of variability within the data just because we are subsampling in the lab. So I think, yeah, so data noise is pretty common to most biological studies and it's not really a problem if the signal is strong enough to be detected despite the noise, or if you can somehow minimize that data noise. So the difficulty with aquatic invertebrate biomonitoring that we see right now is that there's several sources of noise that tend to compound throughout the data collection process. So if you think back to when I was talking about aquatic invertebrates and stream biomonitoring, these same sources of data noise are present there as well, but the signal produced from the stream and river monitoring tends to be very strong because the specific species of invertebrates present in the headwaters, rivers, and streams are very responsive to human disturbance on the landscape. So in other words, we have a high signal to noise ratio when we're monitoring rivers and streams. And this allows for the comparison at the individual stream level, where you can determine if a specific stream or a specific stream reach has been degraded due to human activity. In wetland biomonitoring, the aquatic invertebrate groups don't respond as strongly to human disturbance, and most of those signals are weak. So when we're applying the same generalized or rapid assessment approach that works so well for rivers and streams to wetland sampling, it's not going to allow for the comparison of those individual wetlands because the random noise often overshadows the response. So when it comes to wetlands, we see that we have a low signal to noise ratio. So the big question for us then is what does this mean for the ABMI wetland data sets? And I mean, for me, really, it doesn't mean anything. So Our original intent was not to establish a monitoring approach to differentiate individual wetlands as impacted or unimpacted. Rather, we were looking at monitoring long-term biodiversity changes over time across broad time and landscape scales. And so that's exactly what the ABMI ecosystem health baseline data is going to allow us to do. So by looking at wetlands as groups, either across the province as a whole, segregated by natural regions, or as discrete groups within each natural region, I'm thinking like, counties or watersheds. The random noise vectors from each sampling event, they start to cancel each other out. And so that gives us a reasonable gauge on the status of wetland biodiversity within each region as a whole. So you can see this play out in the hypothetical graph on the right. So if you look at an individual wetland where the green dot represents where that wetland is actually located on the impacted versus unimpacted scale, and the red dot represents the location based on the actual sampling data, you can see that almost half of the wetlands appear to be impacted, even though they're actually doing just fine. But if you look at the 12 wetlands as a unit, so you take the average of the actual wetland status, the green open circle, it's going to be close to the average value of the measured locations of the red circle, the red open circle, because those random vectors of the differences between the actual and the measured values will partly cancel each other out. So really overall, the wetland monitoring system the ABMI has developed it's well suited to that long-term ecosystem health monitoring. But if we want to dig deeper and understand the effects of human disturbance on wetlands at an individual wetland scale, we would need to adopt a more targeted approach that would minimize that noise while at the same time capturing stronger data. So this is something that we've started to explore with a couple of new projects here. So one of the collaborative projects that we're currently working on is the Wetlands Genomics Project, where we explore the use of genomics tools in understanding the effects of agriculture and climate change on prairie wetland biodiversity. So this is a 4-year collaboration between ABMI, Ducks Unlimited Canada, and Innotech Alberta. The project has several end goals, and those include kind of exploring the relationship between agricultural land use intensity and wetland productivity by quantifying aquatic macroinvertebrate and waterfowl populations. And we also want to look at the relationship between wetland productivity and climate change by using pond permanence as a surrogate for climatic shifts. But really the backbone of the project and the focus in the first 2 years here is on looking at the relationship between the number of molecular reads in environmental and community DNA samples versus the relative aquatic invertebrate biomass. So here we hope to develop a system where a more complete aquatic invertebrate data set, along with other taxonomic groups like plants, can be collected more efficiently. And so this approach, if we're successful, will provide a stronger data set by circumventing some of that noise generated by conventional sampling. And it would also increase the taxonomic breadth of the data set, and that would give us a better read on whatever signals are present. So I'm not going to go into the details of the entire wetland genomics project here, but I just want to give you an idea of how the genomics component can help build an aquatic invertebrate data set capable of detecting change at finer scales. So if you want to learn more about the rest of that wetland genomics project, there's a good write-up in the ABMI Wetland Atlas on our website right now. So we know that cells from different organisms and plants present in a wetland all contain DNA. And so as those organisms go about their activities, they're shedding those cells into the environment on a continual basis. And of course, the environment in the case of a wetland is going to be the actual water. So the DNA in those shed cells, it's going to remain intact for a period of time before it starts to break down. So if you were to collect, water sample and filter out those suspended cells, you could actually extract and sequence DNA from, in theory, all organisms living in the wetland, all from that simple water sample. So this is what we refer to as environmental DNA, because you're sampling the DNA present in the environment rather than directly from the organisms themselves. In another process that we call community DNA, you're still going to collect an aquatic invertebrate sweep net sample, but in this case, you would grind up all the bugs in the sample, and then you'd actually sequence the slurry of bugs that were present. So these two processes are actually already in use in several bioassessment programs. But the limitation right now is that they can only give us presence absence information on the different taxonomic groups present in the sample. So although that in itself is quite valuable, it doesn't allow for, good community level analysis. So in order to calculate biodiversity indices, or do any of that ordinal analysis, you want to have the counts and ratios of the different organisms that are present. And so what we're exploring in the Wetland Genomics Project is a way to standardize the sequencing process so that the number of sequence reads from the sample equates to the relative abundance, or in this case, the relative biomass of the different organisms present. So in this way, we hope to have both a more comprehensive taxa list from the sample and the relative presence or relative biomass of those taxa that we can use in the analysis. So it's going to give us a number of advantages over conventional sample collection because the sample collection and processing process is simple. That allows you to collect multiple replicates, and that's going to reduce some of the random noise from the sample collection. It also circumvents the random noise generated from some sampling in the lab because sample is processed as a complete unit. You're going to get a more comprehensive list of all the taxa present, including some that don't actually get captured in your net when you're doing your typical invertebrate sampling. And it's going to give us positive identifications on all specimens, not just the ones that are mature enough to be identified. So really, in short here, you're going to get a more complete data set with less random noise. So we've seen how the current rapid assessment approach falls short when it comes to measuring wetland health and detecting change over short timescales and at individual wetlands. So to address this, another thing we're starting to explore is other monitoring approaches that can better detect small scale change. So what exactly this looks like, we're still working it out, but we have a few guiding goals that we have in mind as we work through this process. So yeah, we're hoping to cooperate and operate alongside some of the other existing wetland monitoring programs to help track that change across Alberta. We want to be able to assess all wetland classes recognized by the Alberta Wetland Classification System, as opposed to just focusing strictly on shallow open water. We want to be able to assess standalone wetland condition and status at the individual wetland level. And we also want to be able to detect change in a wetland over kind of that 5 to 7 year time span. So the approach here is that we're going to use what we've learned from our current data sets. We're going to look at some of the other successes in the literature, and that's going to inform the development of our small scale monitoring approach. So the plan moving forward is that the small scale change monitoring is going to be running in parallel with our current long-term ecosystem health monitoring. So in addition to these two projects, we're continuing to work with the data whenever we can. So far, we've done quite a bit of general data exploration. And the goal there was to really strip everything down to the basics to see how each of those data elements fit together. And so in this way, we could start to build a picture of what might work in terms of data structuring and analysis, what avenues of analysis would be worth pursuing versus those that are likely dead ends. So yeah, there's been quite a bit of work done here. It's been pretty enlightening so far, but we still need to kind of take the successful parts of what we have done and formalize those before we start releasing any results. We've also done quite a bit of work on establishing a reformatted and simplified aquatic invertebrate data download and an associated data lookup table. So we've settled on a system here for distributing unidentified specimen data across the data set. So in the coming months or so, we're going to finalize all of this and we're going to make it available as part of the public data download. So this is essentially the groundwork or the foundation for any analysis moving forward. And it should allow anyone else working with the data to kind of work from a common starting point. So you could expect to see some of this work in addition to the results of our data exploration in the Wetland Atlas. You might see it as part of our data download and on the biodiversity browser over the next year or so. So this is where we're at. I feel like we've barely scratched the surface in what we've done so far, but I guess in real terms, although there's still a lot more work to be done, we have made definitive progress, and also laid some important groundwork along the ways. So yeah, I'm looking forward to the next couple of years as we start to put all of this together and release more data to the public. And with that, we can move to any questions, comments, or points of view.